We're going to go on now to the SSAT session. The SSAT, the first paper is presented by Dr. Ryan Morgan, and that paper is entitled Development of Postoperative Local Tumors and Distant Metastases in Diet, Genetics, and Microbiome Dependent in a Mouse Model of Colorectal Cancer Recurrence. So we'll have the video by Dr. Morgan.
Thank you for the opportunity to present. My name is Ryan Morgan. I'm one of the general surgery residents from the University of Chicago, and I'll be talking about our work from the Shogun Lab. Colorectal cancer is a leading cause of cancer death, particularly in cases of recurrence, and traditional paradigms have suggested that recurrences are due to either incomplete resection or occult metastases left behind at the time of surgery. Multiple studies have raised the possibility, however, that exfoliated cancer cells in the lumen of the bowel may be a source of recurrence as well. In the 80s, Umpleby showed that these cells are present and viable at the time of surgery, and subsequent study found that these experimental cells could cross a watertight anastomosis in a mouse model. Further, the role of these cells is supported by clinical data that shows that a rectal washout decreases rates of local recurrence after rectal cancer resection. And while these studies don't necessarily explain the exact mechanism underlying this phenomenon, they suggest that these cells may be a source of recurrence. These data, along with others, have led us to develop a novel model of recurrence. When the primary colorectal tumor is resected, these exfoliated cells remain behind within the lumen of the bowel, and they're able to interact with the anastomotic microenvironment. Multiple factors can influence this environment, including diet, the microbiome, and the tumor genetic background itself. We propose that the interaction of these factors in the healing anastomosis promotes the migration of these cells to cause both local and distant recurrences. Specific to this study, our hypothesis is that a high-fat diet and its impact on the microbiome would alter the anastomotic environment and change the metastatic potential of these cells. To study this, we've used two separate organoid cell lines, AKPT and KPN, and these two cell lines, as well as other organoids that have been described, demonstrate a wide range of metastatic potential in animal models. However, these tend to be primary mouse models or splenic injection models, and our goal was to introduce them into a surgical model to see if manipulations in the diet or microbiome could impact their propensity for metastasis. The model we used involves giving mice 6 weeks of either a high-fat diet or a normal chow diet preoperatively. We then create a colonic anastomosis in the descending colon, and on postoperative day 2, we give cells an enema of either AKPT or KPN cells. We then serially sacrifice the mice, focusing mostly on postoperative day 21 and 56, and we collect samples for analysis. Our first goal was to show that these tumoroids or organoids were able to form tumors within our model. Here you can see both on gross appearance and H&E tumors in the colon, liver, and lung in AKPT mice with our model. We were able to confirm that these were AKPT cells by performing in situ hybridization for LGR5, which is a colonic stem cell marker. We were able to do the same for KPN, showing that both of these organoids could form tumors within our model. When we looked at tumor formation rates by 56 days, we found that there was a significantly higher rate of AKPT tumor formation with a high-fat diet compared to mice that were given a chow diet. KPN mice showed no significant difference based on diet. However, interestingly, they had an opposite trend towards higher rates in a chow diet. In addition to their higher rate of tumor formation, I noticed that AKPT mice given a high-fat diet were often dying before their planned sacrifice date due to large, bulky tumors that are found on autopsy at the anastomosis. We were able to show that these mice had significantly decreased survival compared to those on a chow diet. And interestingly, we did not see any similar trend in KPN mice, suggesting that this response to diet was at least in part based on the organoid's genetic background. We then wanted to test which factors within the anastomotic environment led to increased AKPT tumor formation. We hypothesized that this effect was likely mediated by the microbiome. To test this, we used fecal microbiota transplants from conventional mice to germ-free mice. We again, in this experiment, had a control group of germ-free mice that had not been given any FMT, as well as a group that had been given FMT from high-fat diet mice, and a third group that was given an FMT from chow diet mice. And all of these mice then underwent our model and were sacrificed on postoperative day 56. When isolated in a germ-free mouse model, the high-fat diet-associated microbiome significantly increased the rate of postoperative tumor development. In the control group and the Chow FMT group, there was a low 10 to 12% rate of tumor formation, with most of the anastomoses remaining free of tumor, as shown here on the left in blue. In the high-fat diet FMT group, however, there was a 40% rate of bulky tumor formation, as shown in red. Having shown this impact of the high-fat diet microbiota, we then wanted to test whether it could be reversed with diet manipulation. In a repeat set of experiments with conventional mice, we again had a Chow diet control group as well as a high-fat diet group, and this time we introduced a reversal group in which mice were given 6 weeks of a high-fat diet followed by a Chow diet reversal period preoperatively. Following this diet reversal period, mice underwent our model. We began with a 2-week reversal period and found that mice in the reversal group continued to have poor survival and high tumor rates similar to that of the high-fat diet-fed group. We then extended this to 6 weeks, however, and the reversal group experienced a more significant increase in survival and a tumor rate similar to Chow-fed mice, suggesting that this response to diet and this reversal of phenotype was time-dependent. We then set out to analyze the stool samples from these reversal groups, beginning with 16S RNA analysis of the microbiome composition. Each bar here represents the complete microbiome of an individual mouse at early time points when exfoliated cells will be present within the lumen of the bowel. You can see clearly that there's a derangement of the microbiome in high-fat diet mice. In particular, there's an overabundance of Allobaculum, shown in orange, as well as a bloom of pathogenic bacteria such as Proteus, E. coli, and Shigella on postoperative day 7, all shown in red. Most importantly, however, we can see that the reversal group very closely mirrors the composition of the Chow group, both in its baseline composition and in its response to surgery, suggesting that the reversal in phenotype we saw may be due to changes in the microbiome. So to summarize where we are so far, we were able to show that a high-fat diet was associated with increased tumor burden and decreased survival for mice given AKPT cells, but not those given KPN cells. We were able to show with germ-free FMT experiments that the high-fat diet-associated microbiome was necessary for promoting high rates of postoperative tumor formation. And lastly, with diet reversal experiments, we were able to show that preoperative diet manipulation reduces tumor formation in mice, suggesting that there may be a clinically relevant role for diet rehabilitation in colorectal cancer treatment. I'd like to thank all of our funding sources, including SSAT and ASCARS. I'd like to thank also all of the members of the Shogun Lab, as well as all of our collaborators across the University of Chicago. I'd be happy to answer any questions.
Ryan, thank you very much. And we're going to discuss your paper in 1 second. I just want to tell people that the poll from the American Hernia Society papers is now up. You can vote while we're discussing this paper. Please vote. Ryan, that was a terrific paper. And I have to just say that, we learned in the past from the work of Turnbull and others that there were factors which affected later metastasis. The no-touch isolation technique of Turnbull was important in telling us not to handle the tumor. We know about drop metastases and shed tumor cells when you handle, but this association with diet is very interesting. How do you suggest that we implement this in the care of our patients who are going to have colorectal surgery? Should we put them on a special diet before, and for how long?
Thank you. First of all, thank you to GlobalCast for the opportunity to present this morning. And it's a great question of how exactly we can translate this to some sort of clinically relevant role for diet manipulation. I think that it'd be difficult to compare exactly the metabolism of mice versus our colorectal cancer patients, and it'd probably be a longer period than what we saw for the mice. However, I think it would be worth studying whether we can give patients a prescribed diet preoperatively that may improve outcomes. I do think that some of the data from our institution has shown that a combined mechanical and antibiotic preparation preoperatively is able to decrease rates of recurrence, and I think that suggests that altering that microenvironment around the anastomosis is able to impact the rates of recurrence. And I think that diet manipulation would play a role in that. The exact length of time, it needs to be studied though.
So do you plan to do this in humans with a prospective study?
We don't have firm plans for that right now, but it is something that we have discussed.
Should be able to do that. Dr. Rosen, any comments?
Yeah, I mean, listen, first of all, Ryan, that's great work. I mean, that's a lot of time. That's a lot of effort. I actually, and it's well done. I mean, one of the things I love about basic science research is it's very well controlled. You have appropriate positive, negative controls. And, you know, and then you get some interesting data that you kind of try and get to figure out. So I was hoping maybe you would expand for just a minute on, like, why would you hypothesize the difference between the AKPT and the KPN based on, you know, diet modification? Is it just a model issue? Is that perhaps a barrier to then understanding it in patients? How do you kind of forecast that for your next steps?
Thank you. So kind of our next steps, what we're working on now, is looking at comparing the two organoids and how they respond to different metabolites that are produced by the bacteria. And we think that that might be the link, explaining why there's a differential response between the two. So specifically what we're looking at is secondary bile acids and deoxycholic acid metabolites like this that are exclusively bacterially produced. And what we see, at least in our preliminary data in vitro, is that there's a bit more of a response from the AKPT cells and the KPN cells in proliferation in response to exposure to these secondary bile acids. What that exact link is as far as pathways, that remains elusive for us. I think one thing that we have hypothesized is it could be related to something like Wnt signaling with the AKPT cells having knocked out APC signaling. That's would be one difference between the two organoids, but we haven't exactly nailed that down yet. One of the other steps that we want to do is take the tumor samples from the mice from either organoid, and we can see with you know, RNA-seq, different things like that, whether there are different pathways that are up or down regulated, and if there's any differences between the two.
Great stuff.
All right, so I think not everyone voted, but we've got a good number of people that have voted. So it looks like from the final poll result that age exacerbates inequity in telemedicine won as the most impactful paper in the American Hernia Society runoff. So congratulations to Connie. You'll be going to the finals later at the end of the hour, and then we'll find out who is the best of the best. Do you want to say something?
No, that's
Yeah.
terrific.